Meiosis 1. During interphase inside the nuclear membrane, the chromatin is preparing for the process of meiosis by replicating its DNA. Meiosis 1. Prophase 1. The chromatin form chromosomes in the shape of an X, and they come together in homologous pairs, called tetrads. The chromosomes are now diploid. Synapsis begins, and the tetrads cross over and exchange their DNA. Metaphase 1. The nuclear membrane disappears, and the centrioles align at the poles of the cell. After this, spindle fibers attach to the centromeres of the chromosomes. Lastly, the pairs line up along the equatorial plane of the cell. Phase 1. The spindle fibers start to shorten, pulling the homologous pairs apart. Then, the sister chromatid are pulled towards the poles by the spindle fibers. Each pole ends up with a complete haploid set of chromosomes. There is one member of each pair now at each end of the cell. Telophase 1. The spindle fibers and centrioles are no longer needed, so they dissolve and the cell starts to pinch, causing a cleavage furrow. Then, a nuclear membrane reappears around the sister chromatids. Cytokinesis 1. Disjunction occurs, and there are now two daughter cells with sister chromatids. Meiosis 2. Prophase 2. The nuclear membrane disappears, and the centrioles align at each pole in both daughter cells. Metaphase 2. The spindle fibers attach to the centromeres of the chromosomes, and they line up at the equatorial plane in both cells. Anaphase 2. The spindle fibers contract yet again, and the sister chromatid are pulled towards the poles in both cells. phase 2. Both daughter cells start to pinch to form a cleavage furrow, and spindle fibers in centrioles dissolve. The cleavage furrow is now forming. A nuclear membrane reforms around the sets of daughter chromosomes. Cytokinesis 2. There are now segregations between the 4 sets of daughter chromosomes. Meiosis 2 is now complete. The result is a set of 4 daughter cells that are genetically different.